Can I see that?
These
that
are
your
maps.
Oh, those are maps.
Can you put them with the maps?
We can give that. Okay, yeah. I think our DNA testing or our DNA barcoding setup is pretty unique in that they were able to pack everything up into 2 suitcases and do it in the field.
The Bento Lab laboratory is a gadget that has 4 major components on it. It has a thermal cycler where you can perform polymerase chain reaction, or PCR, which is essentially amplification of DNA. And then it also has a microcentrifuge in the center. And then it has on the left-hand side a trans-illuminator portion on which you can place gel electrophoresis unit. So you can do the whole PCR process on this gadget, and it's portable, and which means you can bring it anywhere with you.
And tell me that the pictures are okay, because once we take the bug, there are no more pictures to be had.
So 75 microliters, and this is the range that this micropipette can do.
It goes into each tube, but then for the negative control, it's actually, the 1.5 is water.
Okay, so write DNA insect.
So yesterday at the rainforest, we captured an insect and we immediately extracted DNA from it. This is a 20-minute process. So put the insect into the tube and then added DNA extraction reagent, incubated it at 95 degrees for 15 minutes, and then added neutralizing buffer, and eventually this acts as a storage buffer for the DNA. And so we have that DNA, and we have the positive control, which is Drosophila melanogaster. We already know what it is. We will run all the reaction in parallel. So we have our positive running, and then we have our unknown insect running for this reaction. The next step here is to run a polymerase chain reaction, or PCR. And for the PCR, one of the things that we need is what we call primers. These primers will amplify a specific region on our DNA of interest. And so that region that will get amplified is known as the hypervariable region. It will help us identify what this insect is.
We have dissolved the tablet, and now we are ready to heat it up for 1 minute in the microwave. We're gonna use Kim White here as our cap, top here, just a little bit. So the gel solution provides the ions that will be able to drive the DNA in the electric field from the negative to the positive end. DNA is negatively charged, so you set up your samples on the negative end of the field, and then it will run over time for about 30 minutes. It separates out, and that's why you need a solution in there.
So what we're looking for is a little band that says, yep, you have DNA.
Yep, that's about right. So then we'll take it out and swirl it.
That's looking pretty good.
Yeah.
So as a part of our work, we were super lucky to have our bird expert ID a lot of the bird calls that we found. However, we didn't have someone who specialized in insects or frogs that we could reach out to. So we've been using iNaturalist, which is basically a crowdsourcing platform for doing biological IDs. Other users of the site will be able to listen to our recordings and submit possible identifications. So we can use the expertise of the community, sort of leaning into like the citizen science thing that we've been focusing on as a team.
We got the results from Casey, and based on Casey's result, we sorted those snippets based on species names. And for example, this is 3 snippets that Casey identified as the sound that scopes out, and we looked at it in Adobe Audition, and you can see. So here's something very interesting, and I'm going to play it. It's very brief. It's very low pitched. Ooh, ooh, and this is owl, and it's a very cute owl, and we are very happy that we catch the owl in our recorder. Yeah.
I was initially bummed, but mostly grateful to have been part of something this big and something this cool. You know, when I walked into this, I never thought I'd actually be the one going out, flying the drones, doing the big things here. So we did a lot of good work, and I'm proud to have been a part of this.
Just appreciating having worked with such a skilled group of people that are multidisciplinary, and I really do think and hope that our paths will cross again.
I'm super proud of the team because the team did a great job, and I can't say how proud I am of Martin, Professor Broke. Because, you know, the point isn't as much to win as to have the experience and to participate in a competition.
But particularly in engineering, what doesn't get well documented is the fact that almost every success has many, many, many failures behind it. And the more failures you have, often the better the final success, because it means you've tried more different things. And so I tend to tell students when they're trying something new, now you know it's probably not going to work, so what are you going to do when it doesn't work? And it's like, oh, not boo-hoo, oh, let's give up. It's, oh no, well, let's study this. I never go into these competitions expecting to win. All I ever hope to do is compete. My main mission is educational. I'm really not in this to win the competitions. I'm in this for the generation of ideas and the chance for students to get involved in something authentic and meaningful that might have an impact.